Yeah,
and then I will go ahead and introduce our speaker for today for the GGA seminar. And this is Dr. Nathan Crook. And Dr. Crook is an assistant professor here at NC State in the Chemical and Biomolecular Engineering Department. So Dr. Crook did a bachelor's degree in chemical engineering, which he received from California Institute of Technology. He then moved to a PhD at the University of Texas at Austin under Dr. Hal Alper, also in chemical engineering. Went on to do a postdoc at Washington University, St. Louis School of Medicine. This was in pathology and immunology with Dr. Gautam Dantas. He then came to NC State in 2017, and since he's been here, has been incredibly successful. We were lucky that we were able to bring him here. He's received an NSF Career Award and an NIH New Innovator Award. His lab, he has 10, as he says, amazing graduate students and also 3 amazing postdocs. So we're all very excited to hear what he has to say. Thank you.
Well, thank you very much. That's a very kind of, sorry, I'm getting Zoom figured out. I can figure out Zoom, I think. Okay, that's online, that's over here, we're good. Well, I appreciate you all being here, taking time out of your Monday. I know after the weekend, everything is always crazy, but I'm gonna be here talking to you about the work that my lab's been doing since over the past almost 6 years now. I wanna give a shout out before I get started to all of the people that have done the work that I'm gonna tell you about. Well, actually, a lot of people that did the work are no longer in the lab. They've since moved on to bigger and greater things, which is the great tragedy of being a professor, I think. But we have a few folks who are currently in the audience, currently here, who are continuing on this work. I want to also thank my department, right? This is a GGA seminar, but I'm in the chemical engineering department. We're celebrating our 100th anniversary this year. So you invite all the famous alumni of our department back, and it's a really great opportunity for the students to say, oh, I can be the CEO of whatever cool company that is. So it's been a great place. And then I also want to thank, I'm a cluster hire, right? So I'm going to thank my cluster as well. We're a great group of people. We interact regularly and we're good friends. So it's been a great thing for a young professor like me to get started. Okay, last bit of logistics, disclaimer. We have, NC State loves to do invention disclosures. So they encourage me to do that too. I'm going to be wearing my professor hat today, so I'm going to be critical and entertain lots of hard questions about this. So if you have anything that says this isn't going to work, that's totally fine. All right, our gut microbiome, right? We've heard a lot about this, and so from an engineering perspective, we like to give out numbers, right? Our own human genome has about 23,000 genes, 10 to the 13 cells, 70 kilograms of biomass, give or take. But in our gut, you have orders of magnitude more biosynthetic potential. Similar numbers of bacteria and fungi as there are human cells in our body, and not an insignificant amount of biomass. So if you've done a fermentation and measured the biomass in that fermentation, 400 grams of biomass is enough to do a lot of chemistry with. What currently our microbiome does with that is to provide about 10% of our daily caloric intake from fermentation of dietary fiber. It produces vitamins. But what's interesting about the microbiome as an organ is that you can change what it does. Now, normally this develops sort of semi-randomly, but you can change it artificially using FMTs or probiotics. So it's one of the few organs you can actually engineer its function. You can perform chemistries that human cells can't perform. And among the ones that are in your gut, some of them are actually easy to engineer. So why not actually engineer these bioreactors that we have in our own body? And I'll talk a bit about what we think we could do with this. This is sort of the 30-year vision, right? So take that with a grain of salt. So if you think about medicine, here's how it tends to work, right? You contract a disease, it takes some time before you feel sick, right? Once you feel sick, then you go to the doctor. If you're lucky enough to have a doctor around, they'll tell you what's wrong with you and they'll prescribe a treatment. Also, if you're lucky enough, there's a facility that produces drugs nearby or has shipped drugs to you, so you can take those drugs. You feel better, right? This is how medicine works. It's really, really great. That's why I think most of us are still here today. In some places that doesn't happen though. There are some, for example, availability of doctors or synthesis is the thing that we're going to focus on here. What if you could have a probiotic or a food culture that was sort of passed down over time? You could take that microbe, and then once you have a disease, right, that microbe in your gut could sense whether that disease is present and then produce the appropriate molecule in response. That way, you don't even notice you were sick in the first place. This could go from infectious diseases, it could be inflammatory bowel disease, colon cancer, anything sort of gut related. So this is sort of the 30-year dream. But treating diseases is only half the story I think we could do with our own gut microbiota. Human metabolism is limited, and it's limited in ways that we don't fully appreciate because we're so used to these limitations, right? So for example, glutamine, our own body can make, right? If we don't have glutamine in our diet, our body makes it for us. Lysine is not that way. If we don't have food with lysine in it, we die. Glucose is something our body can make use of, but a polymer of glucose called cellulose is not usable by our own body, right? So if you're unlucky to not have glucose or other types of sugars around, you won't be able to survive. Not only that, there are vitamins that our body needs. They're called vitamins because we don't make them by our own body. Vitamin A is one example of a deficiency that's causing big problems in the developing world. There are molecules that nature makes that are antithetical to our survival. So phytic acid is one example where it chelates iron and prevents our own body from absorbing it. So if plants make this to get themselves iron, but phytic acid is a big problem for us as well as for animals. And finally, there are molecules that nature makes that are actively trying to kill us, right? These are all oral molecules, and yet our body has no way to deal with them. So what if we could use the microbes in our gut to change this? What if we could use our microbes to maximize calories and nutrients during growing years, synthesize vitamins required if you're growing a person inside you, and then as you get older, moderating that energy intake to a healthy level, right? So these are all things you can think about if you have an engineerable bioreactor inside your body. So this is the dream, right? You have a bioreactor, you can control the inputs. Dietary fiber is our input, as well as host mucus. But host health markers can then be sensible by microbes to turn various metabolic pathways on and off to yield value-added biomanufacturing inside your gut. So this is the dream. This is what I think this is a similar picture to what I showed when I was interviewing. And so, but we need to actually have engineerable, quantitative, controllable things for this to work. So how are we going to get there? First, we have to be able to engineer the microbes that are present. We have to be able to sense, perhaps sensing disease is one thing you might want to be able to do. You have to make sure that productivity of the thing that you're engineering is controlled, right? And producing something that might be valuable. And lastly, you might have control over the abundance of that microbe. These are all unit operations. The chemical engineers love the word unit operations in your gut that you want to get right. You don't go straight to the goal. You build these up so that you can then, once you have them built up, do whatever you want. Okay, so we'll go through all of these. First is ability to engineer. And the chassis we're using in this work is Saccharomyces boulardii. Saccharomyces boulardii is a promising probiotic, but it's also weird, as a strange choice, a non-traditional choice. The reason why it's non-traditional is that if you sequence the metagenome of your gut, the eukaryotic gene fraction is very, very small. About 0.1% of the genes in your gut are eukaryotic. Yet, eukaryotes are much larger cells on a per nucleotide basis than bacterial cells. If you do a rough back of the envelope calculation, the mass fraction of eukaryotes in your gut can be as high as 10% of your total gut biomass. So it's an interesting idea. And to support this, people have taken pictures of, for example, the mouse gut. This is anti-yeast antibodies staining the mouse gut. This is the mucus, or the epithelial cells secreting mucus, and these yeasts live on that mucus layer. Now, Boulardii is a yeast. It's sold over the counter. If you've drank a kombucha ever, likely it has Saccharomyces boulardii inside it. You can find this at your local pharmacy. It has some properties that have been published, but the mechanisms are not quite well established. Anti-diarrheal, anti-pathogen, it improves gut barrier integrity. But for us, what's interesting is that it's very genomically similar to a yeast we know a lot about, which is Saccharomyces cerevisiae, and it grows at 37 degrees Celsius. So our goal for our lab was to sort of develop engineering tools for SV and to deliver biomolecules to the intestine. So the first thing we did is we took yeast promoters, cerevisiae promoters, put them into Boulardii, right? We already have this toolkit of cerevisiae parts. Let's just see if Boulardii can use them. And the answer was yes. These are all of the promoters that we had access to from cerevisiae. We put GFP at the other end, and we see a graded level of gene expression. So we can turn genes high or turn genes low. The one exception to the rule that they work the same was this promoter called PALD6. This promoter was very, very strong in Boulardii, but it's rather weak in cerevisiae. So let's use this part library to do something useful. We talked about vitamin A in the first slide. Let's talk about beta carotene. Beta carotene is a precursor to vitamin A. It's sort of this molecule here. 12 micrograms of beta carotene is 1 microgram of retinol, so there's a dietary equivalence there. But vitamin A deficiency is a huge problem, but also carotenoids have this anti-inflammatory, anti-cancer property in the colon. There's been studies that show all-trans retinoic acid, which is a derivative of beta carotene, can attenuate colorectal cancer. So here's an example of a mouse colon that has treated with vehicle or all-trans retinoic acid. You can see the colon polyps in this study were lower. So production of carotenoids in the gut could be valuable for not just absorption of vitamins. So what we did is we looked at the metabolic map toward beta carotene. This is a well-known metabolic pathway in yeast, where you take yeast's sort of native sterol synthesis pathway and you upregulate it. You express an HMG1 gene and you overexpress 3 genes from Xanthophyllomyces dendrotis, CRIT-E, CRIT-YB, and CRT-I. Those 3 genes can bring it into beta carotene. This is a good example for metabolic engineers because there's some interesting trade-offs that are present in this pathway. For example, HMG1 brings flux into this pathway, but it also does so at the expense of maybe other molecules the cell might want to make to grow. Also, CRIT-YB produces the thing that you want, but also this other thing that you don't want. And so what is the right answer in terms of how strongly to express these promoters? So being engineers, what we decided to do is try all possible combinations of promoters. And so the way we did this is we took each individual gene and we had about 9 promoters that we were saying these might be useful promoters to try. They were strong, they were weak, they were middle. And what we did is, this is sort of the cloning reaction. I talk about this because it's kind of a cool way Scott figured out how to do this. And we do a BSA1 based sort of Golden Gate assembly in 1 step here. Now that generates 9 different vectors in 1 pot. If, instead of putting that into E. coli, he directly PCR'd that mix. So there's no E. coli step here. Once it was done, he PCR'd it. And then he got 9 different amplicons. He did this not only for CRIT-E, but also for CRIT-I, CRIT-YB, and HMG1. So we have 9 to the power 4 possible combinations of things we might want to be tested. Once those all get assembled together, yeast is really good at putting them into a single vector inside the yeast cell. There's no cloning, there's no E. coli step. You just co-transform all of that in, and yeast combination is so good, you'll get colonized. So he got this plate, and if you look very closely, you can see each colony has a different shade of orange, the different level of carotenoid production. Now, Scott and Dennis then poured over this plate and other plates and got 28 different isolates of this beta carotene polarity that we were interested in. And so we were able to then, through our collaborators, figure out which one had the highest beta carotene production. We took one of those, I think that was isolate number 23, and we put that into mice. Before we went straight to mice, this is again something great about NC State. We have a germ-free mouse facility in our vet med campus, so we can just give them strains and they can put them into germ-free mice for us, and we can do science that way, which is super cool. So we gave germ-free mice wild type Boulardii. We also gave conventional mice 2 different types of antibiotic treatments. These were either a sort of a high dose antibiotic or a low dose antibiotic. It was a combination of different antibiotics. And the notion of the antibiotics is that it depletes the bacteria so that the yeast can survive better. In this case, what we saw was that over the course of 30 days, mice that had just yeast in their gut could survive that 30 days. And also the yeast was not washed out over that course of 30 days. By talking to the facility people, they mentioned no adverse consequences of having an entirely Boulardii gut microbiota for these mice. I'm not saying I'd recommend for a person, but it's useful to know that there was no overt pathogenesis here. And for the mice that were conventional and were antibiotic treated, the colonization seemed to correspond to how many antibiotics we gave the mice. So this tells us one important thing is that this yeast doesn't colonize very well. It's not a permanent resident of the gut, so it's going to come up later. So we said, okay, if we're going to have any hope of seeing beta carotene in a mouse, what we need to use is a germ-free mouse. That's our baby step. We want to go in baby steps to make sure that we know what we're doing. So we gave this engineered yeast to these germ-free mice, and that is shown here, where we have a mouse that has given either wild-type yeast in black or this beta carotene producing yeast in orange. In this graph, we're showing firstly the colonization load. So the wild-type yeast colonizes to a little bit higher than the beta carotene producing yeast. We thought this made sense because beta carotene is sort of a metabolic burden on the cell. It's going to survive less actively in the gut. However, the beta carotene levels showed that the beta carotene producing yeast had significantly higher beta carotene levels in the feces than the wild type strain. There was some residual beta carotene here because of the diet. The diet has carotenoids in it. So we were able to detect that, plus whatever we gave that mouse. And it was stable over the course of 2 weeks, which we thought was quite interesting. This was on a plasmid. We were putting all of these strains, these genes on a plasmid in yeast. They weren't integrated into the genome, and yet we got 2-week-long production of beta carotene in this germ-free mouse model. So that was quite exciting. And it was actually, if you integrate the beta carotene delivery over the course of that 2 weeks, it was providing about half of the daily recommended value. So if all of that beta carotene in the feces was making it into the body, that's what would happen. Now, we don't think it's getting into the body. Yeah.
What kind of plasmids were they, centirs or 2 microns, sort of like high copy or low copy?
They were high copy, 2 microns, yeah. What was interesting about it, when we took these fecal samples and we plated them on both non-selective and selective plates, the non-selective plates, they still had they were still orange. There was no detectable. But we thought what would happen is you would have plasmid loss, and then the strains that had lost the plasmid would dominate the gut afterward. And we didn't see that. We're still confused as to why. All right, so we can engineer a thing to do something that could be interesting, right? Beta carotene is one thing. But what if the thing that you want to treat is something you don't want to have on all the time, right? Vitamins are a good example of something you might want to have on a lot of times, but maybe not all the time. And so in order to see how we're going to sense something, we're first going to go into how yeast mate. I think a lot of you in this audience know how yeast mate. Some of my audiences don't know how yeast mate. But there's 2 mating types of Cerevisiae, an A type and an alpha type. They both secrete hormones that the other one can detect. Once they detect, they shmoo, that's the term for them mating, and they form an A alpha diploid. This can then sporulate and mate again. But the way that they sense that mating hormone is through the use of a G protein coupled receptor. G protein coupled receptors are a very well-known class of sensory molecules in eukaryotes. They have this transmembrane domain that, when it's activated, causes the G alpha protein to cause downstream signaling into gene expression. So yeast has 2 of these natively, only 2. One is for mating and one is for glucose. Those are the 2 things it cares about, mating and glucose. We, on the other hand, have 800 of them, right? They're in our nose, or our tongues, they're in our gut. They're sensing things that our body cares about. And so what's interesting about these is that they're the same class of protein in these 2 cells. Now, we're not the first people to think about putting GPCRs into yeast, but we collaborated with people who have done that. So we're going to put them into Boulardii today. We collaborated with Emil Jensen. He's at DTU Biosustain, and he's really good at taking human GPCRs and getting them into Boulardii. What we did is we take that human GPCR and make a G alpha chimera. So half of this G alpha protein is a human G alpha, and half of this protein is a yeast G alpha. And that seems to be a nice trick. You can determine where that chimeric boundary is. You can tune different parts of the mating pathway such that once it's activated, the FUS1 promoter makes GFP. This is normally active when it senses alpha factor, or the mating hormone in yeast. This one can then be turned on by an exogenous ligand that you have control over. The thing that we had trouble with, though, is that Boulardii can't sporulate like Cerevisiae can. Cerevisiae, you can make these sort of haploid cells that can respond to mating factor. Boulardii, on the other hand, is a diploid, and it has a defective sporulation locus. It has stop codons everywhere in its sporulation locus, so it can't sporulate anymore. What we decided to do to get around this problem was use CRISPR-Cas to just make it a MAT-AA diploid cell. Instead of convincing it to sporulate, we kept it a diploid and just changed its mating type using gene editing, which is a fascinating thing to do. So with that, new cell is now responsive to its cognate mating factor. Once you've changed its mating type, it's able to sense alpha factor like normal in the culture. Okay, so now we have our strain that can sense things. Let's put cool GPCRs in front of it. One of them, what we did is a P factor, that's Schizosaccharomyces pombe's mating factor. It's another kind of yeast, it's a fission yeast that has its own mating factor, and now we can sense that. There's a human GPCR for adenosine and melatonin, and in both cases, we were able to get Boulardii to sense those as well. So I think this is a cool platform for sensing other things that you might care about. We have an ongoing NIH proposal with Emil to sense Candida's mating factor, right? So Candida makes mating factors as well. So you can think Boulardii could be a nice sentinel for the presence of mating competent Candida in our guts. Okay, we've gone through engineering and sensing. let's talk about producing something that we might care about. The thing that we have cared about for a while in our group has been Clostridium difficile. It's a really big problem, and it's a collaboration between some folks in my department, Steph, Carol, as well as Scott Magnus at UNC DME. We also have Casey Terrio, not shown here, but we're collaborating with Casey as well. So C. diff infection. A lot of you know why this is such a big issue. Some of you may not. The reason why this is a bacterium that infects your gut, it tends to, it has a spore form and a vegetative form. So the spores can get into your gut through a variety of ways. A lot of people have C. diff in their gut already, but they just don't know it. But if you get treated with antibiotics, there's a lot of niche space available in your gut. There's a lot of nutrients because those other bacteria have died. Now, these spores germinate in guts that have been treated with antibiotics. Once they germinate, this vegetative form produces at least 3 different types of toxins that we know of. One of them is one of the important ones. It's called toxin B. But that toxin that it secretes destroys your gut lining. It makes fluid come from your body to your gut, from your gut to your body. It can lead to sepsis and death. And the other issue with this is that while it's sensitive to antibiotics, it has a high relapse rate. So 20% of the time, it'll come back. And that next time it comes back, your probability of being treated is less than 80% at that point. If you relapse, you're likely to relapse again. So it's this cycle of going in and out of the hospital, debilitating pain, lots of medical costs. All right, so this toxin. The toxin is the thing that causes diseases. So let's target the toxin. This toxin is a fascinating molecule. This toxin is a huge protein, 300 kilodaltons. And most of this toxin molecule is involved in getting this thing from outside our cells into our cells, right? The C. diff secretes it out into the extraintestinal milieu, and then it's trying to get this orange domain, this glycosyltransferase domain, into our cells. The way it does that is it binds to a receptor. This gets endocytosed. Now, the acidification of the endosome causes conformational change in the toxin, makes a pore in your endosome, and self-translocates itself out of your endosome. So it escapes once it realizes it's being acidified. It cleaves off the warhead domain, this GTD, and now this glycosyltransferase domain has one job, is to add a sugar at a very specific place to our Rho and RAC GTPases in our cells. And when it does that, these get inactivated. And once those proteins are inactivated, the cytoskeleton of our intestinal cells breaks down. So it's a very fascinating protein, but it causes a big problem. So our goal here was to develop chemical inhibitors of this GTPase, this GTP transfer, this glucosyltransferase domain. All right, so how are we going to do this? Steph had a great idea, and that was to take beads that had been coated with, so we were going to use peptides for this. We think peptides are great because they're like small molecules in that they can easily diffuse, but they're like proteins that they're programmable, and then you can change their sequence in a way that you could have a tight binding to your protein of interest. So we're really good at peptides. We wanted to try a peptide-based drug. They coated beads, and each bead had a different peptide on its surface, but many different copies of that peptide. What we did is we labeled that toxin in red. So we had a red labeled fluorophore attached to that toxin. Now, if the toxin can bind to the bead should turn red. But it can bind in multiple orientations. It can either bind to the active domain, where you have this sort of mouth here, which is where you want that peptide to bind. But it can also bind somewhere else in that toxin and not give us the activity we're looking for. So to overcome that, what we did is co-incubate with a substrate analog called UDP glucose fluorescein. So if that's able to get into your toxin, then you know that if a bead is both red and green, it's bound to that toxin, but in a way you don't really want, because you can still have that substrate binding in the active site. So we looked, we used microfluidics. Yes, we used microfluidics here, got a bunch of beads, labeled them with a bunch of different peptides, and we sorted them, ones that were only red, but not red and green. And Ethan helped us do this, which was super cool. And you get sort of data like this. What we had to do is tune the amount of UDP glucose incubation, because the toxin itself is rather large. It can't diffuse into your bead very well. You can see sort of a halo of toxin. This is toxin A here on the surface, but the UDP glucose gets right in. It can get in. There's little pores in these resin beads that it can get in quite well. So we had to make sure that was happening. But we found a peptide that we liked. It's called LS5, and we collaborated with the Magnus group at UNC Chapel Hill, and they have this really great system where they can take primary human colonic epithelial cells. So these are organ donors, that have donated their organs to science, and he can take their colonic epithelial cells and grow them in monolayers on a transwell plate. And what happens is they form monolayers and they're electrically insulating. They're so tight that electricity has a hard time passing through that monolayer under healthy conditions. If it is impacted by toxin, however, that monolayer becomes impaired and electricity can pass through. So what we're looking for here is this resistance to stay high over time. So here what we see is that a toxin by itself causes a loss in barrier function. We have LS5 and we have S1, both of which mitigate that toxicity to actually a greater extent than just cells without toxin by themselves. So this is really exciting. We did this and we were really excited. We wanted to publish it and put it into mouse models now. And then Casey told us, toxin A doesn't make mice sick, weirdly enough. So this is an example of why collaboration is so important, because Casey knows that the only toxin that really makes a difference in humans and also in mice is toxin B. If we wanted to test this, we have to go to hamsters, and we weren't prepared to go to hamsters at this point. So we thought, okay, let's go to toxin B now. It's just a different letter, so hopefully it should be not toxin B. Toxin B has a similar mechanism of action. And so we did that whole peptide-based screening again, and now we collaborate with Carol Hall, who's an expert in electrodynamic simulation. She can take the crystal structure of the toxin and that peptide and dock them, and then have it wiggle around a bit to look at and estimate sort of almost like a KD or a delta G of binding for that thing on her computer. And so here's what she's doing, where she takes a starting point peptide. What she does is she looks at both conformational changes of that peptide as well as sequence changes. So she can make sequence changes that kind of drive that peptide toward a minimum in our binding energy. She found one that she really liked. This is SB6 here. It's sort of lying on top of this reactive loop of this glycosyltransferase domain. So we thought, okay, this is a nice peptide to try and test. We've done this again. We're doing it for the right toxin this time. Let's see if this peptide works. Now, Carly is really great at enzyme kinetics. She remembered her undergraduate and graduate enzyme kinetics. I didn't. So you're going to see line weaver plots here. What she was able to do is vary the amount of toxin. On this axis, we actually had 3 different variants of toxin B that she tested against. There's different sort of ribotypes of this toxin, as you would. And then vary the UDP glucose concentration and the RAC1 concentration. RAC1 is the target of this toxin. So it's another substrate. There's UDP glucose and RAC1, both of which are substrates of this toxin. And by inspecting sort of where these lines intersect with the different axes, he was able to deduce one important thing was that this peptide was non-competitive with respect to UDP glucose. So it inhibits the toxin, but not via displacing UDP glucose binding. However, it is competitive with respect to RAC1, which indicates we think what this is doing is preventing it from actually contacting its substrate protein. So that was pretty cool. We then went to Scott again and said, hey, here's this peptide it's going to be better this time because we're doing toxin B now. So what he did is he took his same cells and he was looking at toxin B and then various peptides. So the peptides without toxin B, this is toxin B by itself, shows that reduction in resistance. Peptides alone without toxin show very little reduction in resistance, which means that these peptides themselves are not toxic, right? You don't want these peptides themselves to be toxic. We have a negative control peptide with toxin B, which indicates that not any old peptide would work. And then we have one of the best peptides, peptide number 6, delayed the onset of toxicity with toxin B. So this peptide isn't working as well as our toxin A peptide was, but it's something which I thought was pretty interesting. What we're currently doing is further iterating on the computational side of things to get this to work a little bit better. An exciting area of iteration, which is new and upcoming in the lab, is that we've surreptitiously, surprisingly, or I would say accidentally discovered that yeast has the same targets of these toxins that humans do, right? So humans, sort of the Rho and RAC GTPases are here, and they're involved in a variety of cellular processes. They're highly conserved across eukaryotes. Yeast has them too. And so if you look at all of these sort of bacterial glucosylating and ribosylating toxins, they sort of interact along this very conserved signaling pathway in eukaryotes. And so we thought, well, if that's true, maybe we can find inhibitors of toxins by just directly looking at yeast survival. If we express a toxin, it should kill yeast. If we express that inhibitor, it should make that yeast grow again. So that was a question. We were encouraged by the fact that human and yeast proteins have a similar sort of structural and pairwise sequence identity to each other. They're not perfect, but they're similar. And people have actually swapped out yeast row A for rack 1, for example. You can do these sort of swaps, and they're functionally equivalent. So what we did is we took all of these yeast toxins and we put them under the control of a galactose-inducible promoter. So this promoter in yeast is off in glucose and it's on in galactose. And what we see is that expression without, in glucose conditions, all of these proteins themselves allow yeast to grow to varying extents, right? This isn't perfect. Some of these are very, very toxic proteins, but in the presence of galactose, they immediately turn off yeast cell growth. The yeast dies right away, which indicates that these toxins that kill human cells also kill yeast, which is good for us. This is data we haven't published yet, but I think it's really cool to see. You have some inhibitors. These are SB, the toxin B peptides, and you also have some other nanobodies we've been looking at. So under glucose conditions, all of these cells are able to survive okay. But when you induce the presence of the toxin, the control cells largely die, but these inhibitor expressing cells allow the yeast to live, which I think is super interesting. You could imagine using this at high throughput to find better and better inhibitors of these bacterial toxins. Now, finding the inhibitor is one thing. Producing it is a whole other thing. Yeast has a very well-known secretion apparatus. It starts in the nucleus, goes through the endoplasmic reticulum to the Golgi body, and then outside the cell. Now, we're lucky in that yeast has been studied for decades as a protein production platform. It's used to make insulin. It's used to make a variety of other different types of proteins. And so what we did is simply search the literature for all possible genes that had been studied for yeast protein expression, and we knocked them out of Boulardii to see which ones gave us the best protein secretion rate. Now, this is that data. Dennis did this, and what she did is she took all these strains with her to Denmark. She had a study abroad in Denmark at Jose Luis Martinez Ruiz's lab. He's got bioreactors. They can run these on. And she made all these knockouts. These are homozygous diploid knockouts, right? Because this yeast is diploid. So she had to make both of them. And what she got was something quite interesting. So some of these knockouts didn't do anything. Some of these knockouts did something cool. And some of these knockouts were very, very good, right? They were producing peptide at much higher levels than the wild type was. We were very interested in these three knockouts here because they were all extracellular proteases. These are all things that this yeast makes that serve to just degrade protein in its extracellular environment. Say, okay, well, yeah, that makes sense. If we had maybe thought about this for 5 minutes before doing this, we probably would have figured that out. So we thought, okay, if these are all proteases, we should just combine all those protease knockouts together to make an even better protein secreting strain. So she was in Denmark at that time, and her ability to do cloning there was worse than it was here. The water was different. It was not working. So she called up her friend Ibrahim and said, hey, Ibrahim, make me all of these combinations of knockouts. So here's that same data just shown in a condensed form. The PEP4 PRB1 double knockout improved production, but the triple knockout didn't. You're like, oh no. I mean, it's still better than wild type, for example, but it wasn't as good as we were hoping for. I said, Dennis, this is totally fine. These sort of things happen all the time in biology where you think it'll work and it won't work, and we can still write the paper. And she's like, no, I have one more knockout I want to try. She had done a surface or supernatant proteomics at Metric and realized that there was this other protease called APE1 that was present in the supernatant of Boulardii. We hadn't knocked it out yet. So that knockout actually does improve secretion relative to wild type. And to this day, we have no explanation for this. When you stack APE1 on this triple knockout, you get a great level of protein expression. This is nanograms per microliter or milligrams per liter, right? So you do that. So this is producing 5 grams per liter of peptide, which is quite nice. And we're hoping to use this strain as sort of our platform for the gut. We don't know if this produces this much in the gut. And we also don't know if it would work for larger proteins. These are things that remain to be tested. And we talked about controlling the synthesis a bit earlier. There's other ways to do it. We did it through the use of GPCRs. You can also do this using promoter engineering, where you have actual binding sites of transcription factors on promoters in yeast. And if you have the right molecule binding to that transcription factor, you either do or do not get expression afterward. This is a classic sort of bacterial controller with TET-R, and they have a nuclear localization signal here so that it works in yeast. Here we have 5 different inducible systems we try. This is copper, IPTG, xylose, galactose, and hydrotetracycline with their cognate inducer. We see that most of the promoters respond quite well to sort of fold induction. This is 23-fold induction with that inducer versus background. But some of them have these sort of off-target activities. And so we thought we should choose the ones that have the highest sort of on-target result there. But what we were interested with the way this galactose was, well, does this actually allow you to sense galactose in the gut? Can we give a mouse this strain and have it light up when in a mouse and see where this yeast actually is? So that's what Dennis did. She took mice that were either treated with antibiotics or antibiotics prolong the colonizations that we could actually see something in this experiment. And then either with or without galactose. And uninduced means no galactose, induced means with galactose. This was a SB strain that had a nanoluciferase expression under galactose inducible control. And you can see in these intestines is that under an unengineered yeast, you see sort of background levels of expression. If it can make nanoluciferase under the control of galactose, but you don't give galactose in the media, you see some lighting up. We think that there's galactose in mucus in the gut. And so we're thinking, okay, maybe it's sensing that free galactose in the mucus somewhere. But then when you give it galactose in the water, you can see this yeast light up. This is mostly in the distal small intestine and in the cecum of that mouse. So it's pretty not very much in the colon. And this is the same data, but just quantified. There's a significant increase in fluorescence after nanoluciferase or after galactose was induced. All right, we're in the home stretch. We're going to talk about abundance now. This whole time has been talking that this yeast doesn't colonize very well. So what can we do about that? The reason why this is hard is that, and I'm just preaching to the choir here, colonization is a complex task, right? A microbe has to go from wherever it was grown. This could be a bioreactor or a food culture, right? Very nutrient rich. There's no other species around to compete with you for nutrients. And then get into a place where it's the opposite, right? Nutrient poor, other microbes are there. There's viruses, there's an immune system, there's flow trying to push you out. So any one of these things could be the problem for this bug. We don't know which one it is, but we had a hypothesis. Our hypothesis was that consumption of the gut mucus is something that a lot of other gut bacteria do very well. Commensal bacteria, not the pathogen, but pathogens do it too, but commensals do it as well. And gut mucus is nice as a food source because it's largely consistent across us as individuals. If I wanted to engineer this yeast to eat a food source, that's going to be different depending on whether I ate tacos or sushi that day, right? But if gut mucus has a largely conserved carbohydrate profile, it might be more consistent across patients. Now, gut mucus, zooming into gut mucus, it's got a protein backbone. It's heavily glycosylated with a sort of tree of one of 5 different carbohydrate sugars. Here are the 5 different main sugars in human gut mucus. And unfortunately, this yeast can't eat any of them. It's not good at eating any of these. Now, GalNAc was actually a false positive in this growth curve. We tried it again and it didn't work. What was weird about this was galactose, right? I've shown you before that Cerevisiae can eat galactose. That was the drug screening part of this. But Boulardi cannot eat galactose. That was very strange to us. It's been published before that this yeast can't eat galactose. What happens is that there's a premature stop codon in PGM2. This is involved in galactose metabolism. It causes the accumulation of what's thought to be a toxic intermediate molecule for this yeast. So we did the engineer thing and we just put that correct codon back in. And lo and behold, when you do this, this yeast is now able to consume galactose. It grows at 30, it grows at 37. It can eat both sugars at this, well, not at the same time, but one after the other, like Cerevisiae can. That was quite exciting. And we thought, well, if it's now able to eat galactose, is that going to lead to a benefit for gut colonization? And the answer was maybe. We had 4 mouse arms here. All of them had antibiotics. One of them had either wild type Boulardi in the sort of lighter color, engineered Boulardii, which can eat galactose in this bluish color, and then galactose. And so what we ended up finding is that this wild type Boulardii with galactose was the worst at colonization. And the engineered Boulardii that could eat galactose was the best, right? These two were significant. There was no real significance if you look at other types of comparisons, but these two were significant. And we think that the reason why this one did so poorly is due to that toxic intermediate accumulation that was there before. It doesn't seem to be that much better than, for example, the Boulardii by itself without galactose. If you squint, and maybe you can see something, but we're not sure yet. And you can look at this over just the fecal material as well as in the intestines. And you see that in the intestines, the engineered strain also does this as well. So we think that mucus consumption of other sugars, not just galactose, could be a useful way here. So we're further exploring that with one of our students in the group. And that is my, what we've done in this talk is kind of walk you through how you might think about production of things in the gut, right? It's a very complex place, but a very exciting place too. We think there's a lot of cool work to be done on the quantitative side to get this a bit more controllable. We think that there's a lot of work that could be done on the application side. Maybe you have a disease you're interested in, and maybe you can use Boulardii. Maybe it's not Boulardii, maybe it's a bacterium that might be better suited for your task. But we think it's a nice place to think about. So before I allow you guys to ask me questions, I want to thank again you for listening and the group for doing this really great work. Thank our funding sources for taking a chance on a young professor like me. And yeah.